All right, so I have a few things that I did not yet mention about peripheral B-cell responses, so I want to talk through those details. After we talk through those details, we are going to go through this problem, which I will hand out right now. It's a problem that is pretty similar to something that is often asked as a way to apply this peripheral B-cell material, but students often don't quite realize that they learned the things that they need to know to answer this problem. So we're going to talk through it and see how the material that you're using in thinking about peripheral B-cell responses could be used in a problem like this. So recall that we learned about ways that B cells in the periphery can become activated. Those B cells are going to become activated with an interaction with antigen, as well as with help from a CD4 helper T cell. Remember that CD4 helper T cell first needs to get activated by an antigen-presenting cell, like a dendritic cell, elsewhere in the lymph node. That T cell can then move to the junction of the T cell and the B cell zone and help out our B cell. That'll happen via a CD40 ligand interaction. And then our B cell will go into the germinal center reaction, start to become a plasma cell, making high levels of antibodies, class switch, do somatic hypermutation, all of the stuff that we talked about last time. Understanding this process is critical for generating antibody responses. And so if we, as immunologists, are generating, say, a vaccine against a pathogen, and we want to make antibodies, we need to remember that this is how an antibody response gets made. If we as immunologists, or as biotechnology people who don't care about immunology at all, want to make antibodies that we're going to sell to use in flow cytometry or Western blots, or that we're going to sell to act on against PD-1 to be the new cancer treatment that wins a Nobel Prize. If we're going to make any of these antibodies at high quantities just to use for something, we also need to remember this process is how we get a B cell to become a plasma cell and make a high level of antibodies. And as a result, there are a few ways that we actually really have to think through this process. So when we think about a B cell epitope, or we think about an antigen that binds to a B cell, what types of biological molecules can be our B cell epitopes? Oh, that was somebody getting a cable, not the hand. Yes. Huh? Proteins can be B cell epitopes. Yeah, Cal. Lipids can be B cell epitopes. Yep, Lee. Carbohydrates. Yeah. Nucleic acid. Really, basically anything can form a B cell epitope. What types of things can form a T cell epitope? What do you think, Kay? Proteins. Maybe some lipids, but that's not a traditional T cell. And this can become a problem. Our B cell might be turned on by, say, a carbohydrate antigen, but for this T cell help business to work, we need to have a T cell epitope, which must be a protein. So how in the world could we get high levels of antibodies against some type of antigen that's not protein? And of course, we also have to make sure we get this dendritic cell step to work correctly as well. This is a real problem. And this is one example of this problem. Here you can see something labeled as toxoid. Ignore the label of toxoid. I should have put a little white box over that label. I don't want to deal with the toxoid right now. However, what you can see is that we might have a B cell that might bind to a polysaccharide antigen. And in fact, this is real life. There are some vaccines that you get where the vaccine is really inducing antibodies to a polysaccharide from a bacterial cell wall. So there are a number of polysaccharide antibody-inducing vaccines. So you can imagine, okay, so our B cell can bind that polysaccharide antigen. This problem is the same problem if this was a nucleic acid. You could imagine a similar problem if it was a lipid. I usually stay away from asking this question about lipids just because everything gets messy with lipids. You could even imagine this if the antigen was a short peptide that wasn't long enough to fit into an MHC class 2 molecule. So if it was a really short peptide that can't be presented on class 2, we got the same problem. You can see that the polysaccharide can get degraded by that B cell. It can get internalized when it binds to the B cell receptor. It can get degraded here. But that polysaccharide is never going to bind to MHC class 2. And so this B cell is going to be out of luck in terms of presenting to a T cell and getting help from a T cell to be able to finish the steps of this response. Thus, when we actually do this in practice, we will usually take our polysaccharide or our nucleic acid or our compound or our short peptide that's not long enough to fit on MHC class 2, and we will attach it to another protein. Typically, that small thing, like the polysaccharide that is shown here, is known as the hapten. And so we'll typically take our hapten and attach it to some other protein. The thing that's labeled as toxoid here, again, it should be labeled like protein, not toxoid. That protein is usually known as a carrier. And very frequently, we use very large repetitive proteins. There are a few that immunologists use a lot here. Sometimes we use one called BSA, or bovine serum albumin. Sometimes we use one called CGG, or chicken gamma globulin. Sometimes we use one called KLH, or keyhole limpet hemocyanin. People use that one all the time. A limpet is a marine creature. If you Google it, it's ugly. And so the idea is that you take your hapten, the blue flower, and attach it to a carrier protein, the red protein. They must be physically attached, because the idea is that when the B cell binds to the hapten and internalizes the hapten, it will degrade both the hapten and the carrier. So you can see the protein and the polysaccharide are degraded here. And now pieces of this protein can be presented on class 2 in order to give that B cell some help. This process is known as linked recognition. And one thing to realize about linked recognition, the hapten and carrier must be linked to make linked recognition work. So you can see that if we wanted to make an antibody response against our polysaccharide, we could attach it to a protein. That can be internalized, and the protein, which is red, can be presented to our T cell. Now our T cell can help our B cell, and our B cell can make high levels of antibody against our polysaccharide. So one thing that you'll notice here is that our B cell and our T cell are not recognizing the same epitope. They're in fact really one's recognizing a polysaccharide, one's recognizing a protein. But because you linked them together, because they were physically bound together, you were able to make it so that B cell could make this response. You were able to allow for linked recognition. So very frequently, when we do this, if you really want to make a response to some kind of hapten, some small molecule that doesn't have a T cell epitope, you will couple that hapten to a carrier. You'll make your hapten carrier conjugate, and immunize the rabbit. For example, the hapten doesn't, by itself, won't really lead to any antibodies being formed. The carrier will lead to antibodies being formed. Here the carrier is BSA. And when you immunize with the hapten carrier conjugate, you will get antibodies to both the hapten, the carrier, and sort of the junction in between them. And then you, in the laboratory, select the B cell making the antibody you want for further use. So you say, aha, I have some anti-DNP B cells. I'm going to select those and use them to be my factories for lots of antibodies. And so if you are an antibody manufacturer who's going to make antibodies to sell, you need to know how to do this in order to make sure that you're getting high-level antibody against, say, your polysaccharide. And if you're a vaccine developer, you might need to do this in order to make high-level antibodies against polysaccharide. Yep, Molly.
This is a stupid question, but this is not physiologic at all, right? Like, this is only, like, scientists do this to make antibodies.
So Molly asked an interesting question. She says this is not physiologic at all. This is only something that scientists do in order to make antibodies. And my answer is yes and no. So yes, if I immunize, there are no situations where a hapten DNP bound to KLH is found in nature. It's just not a thing. And so if you were immunized with purified DNP, you would not make an antibody response. However, in real life, you are often infected with pathogens that have carbohydrates and lipids and proteins and nucleic acids. You could imagine, for example, an infection with E. coli that has some proteins on its surface and has some carbs on its surface. You are going to end up making high-level antibodies to those carbs because they were physically bound to some proteins, because they were physically linked all as part of that same E. coli. So most likely this is happening because our pathogens have multiple types of antigens and multiple types of epitopes in real life. But most of the examples we give of this are artificial. Does that address your question? Other questions? Okay, so now I want us to look at this problem. And this is an old problem I gave out in the past, talking about this exact issue. And you feel like you understand things right now, and you're going to look at this problem and be like, huh? Who? So that's why we're going to talk through it. So it is the year 2050, and your friend has just returned from a vacation to Mars, where he contracted an unknown disease. After a few months of intensive investigation, he finds out that the pathologic agent is an alien organism that causes disease by secreting a polysaccharide that he calls ALF, otherwise known as ALF. In order to better understand the mechanism of this disease, he decided that it would be very helpful to raise antibodies against this. It should be polysaccharide. So he injects a concentrated stock of ALF in rabbits. You could see, if you looked at this question, there might be similar things where it might say small peptide that's not big enough to go on class 2, like a 4-mer, a 4 amino acid peptide, or basically anything that can't be presented here. Polysaccharide is often a go-to, but you can go with a bunch of different things here. So we want to make antibodies. And when your friend measures the antibody titers, he's very disappointed to notice that no rabbit has mounted an immune response to you. But then he remembers you were very knowledgeable in immunology, so he calls you for help. So your first suggestion is that your friend needs to include something called complete Freund's adjuvant, or CFA, which is a preparation that includes mycobacterial cell wall components. And just so you know, LPS or peptidoglycan would work here. So you need to put in CFA along with ALF to generate a high-level antibody response. And the first question is, why do you need CFA? And this is the one that tends to throw people the most with this process. So as you look at the description I've given you of CFA, we can think about what it says to you, what it reminds you of that might help us address this question. So when you see my description of CFA here, what types of things are you starting to think about? Yeah, Melina.
All the stuff that's in the cell wall.
Okay, so those are all things that are involved in the cell wall of a microbe. Okay. Yeah, Jordan.
The things that would be involved in like class 2 presentation from the microscope.
Okay, they might be things that are involved in class 2 presentation, but there's something else they all have in common. Yeah, Amelia. So, not completely. They do have some saccharide components. Alina's thing about the cell wall is the closest so far. Yep, Lee.
So you might need like a pathogen to start the activation process.
They're all parts of the pathogen, and Lee says maybe you need a pathogen to start the activation process. How might you need a pathogen to start the activation process? Yep, Arlene. Yeah, they're all PAMPs. All of those things are PAMPs. And so what's a PAMP going to do? What's it going to do, Jordan?
It's going to, PRRs will like be signaled from the
So we're going to activate a PRR. How does activating a PRR help in this business? Yep, Cal. Exactly. This is helping you with that dendritic cell step. You need this so that your antigen presenting cell can turn on a T cell. CFA is needed so that your T cell gets signal 2 along with signal 1 so that your T cell is activated and that your T cell is ready for T cell help. And in fact, if you looked at that Janeway thing that I gave you guys when we talked about the pattern recognition discussion last week, I told you about there was a paper from Janeway that was a background thing. He actually talks about the immunologist's dirty little secret, which was that you had to add CFA to stuff and nobody knew why for the longest time. And only after we discovered PRRs did this connection finally get made. So in there, if you read that piece from Janeway, he just talks about the dirty little secret of you have to add CFA and we don't really know why. So first, we've got to activate our T cell. And to do that, we need our antigen-presenting cell to give signal 2. So the next part of this process is in B. So you also suggest that your friend should immunize the rabbits with ALF, as well as CFA, which I tell you what CFA is again, and KLH, keyhole limpet hemocyanin, a very large carrier protein that is often used in immunization like this. Your friend heads straight back to the lab and injects his rabbits with 3 separate injections in 3 separate syringes. So one of ALF, one of CFA, one of KLH, 3 separate syringes. And this strategy still fails. And so the question is, why does this still fail? Yeah, Molly.
Because you need your antigen, your non-protein antigen, to bind to your KLH in order to get a response. So if you're getting 3 separate injections, there's no binding happening, so you won't get recognition.
Yeah. Key thing, this is a linked recognition issue. So the hapten and the carrier must be physically linked to each other so that the same B cell can internalize them. You might see versions of this question where I say, you inject 1 into the right leg and 1 into the left leg, or various other crazy things. The moral of the story is that they must be physically linked to one another. They cannot be separate. Sometimes students ask the question, what about the CFA? The CFA is usually given in the same syringe, but it does not have to be physically linked. So the CFA does not have to be physically linked, but you usually give it at the same time. Mostly because it would be a pain to make 2 syringes when you could just make 1. Yeah, Jordan.
The ALF and the KLH.
Yeah, the hapten and the carrier, which in this case would be the ALF and the KLH. Okay, so then the final question, how could the immunization strategy in B be altered so that it would be successful? Justify your answer. How are we going to make it succeed? Yes, Kay. Yeah, and really when I would grade it, I would look for the words like physically linked. Make sure the ALF and KLH are physically linked. before injecting. So that would be ideal. And so now that they're physically linked, we will be able to get this type of internalization of both the hapten and the carrier by that B cell so that the B cell will now make high levels of antibodies. Yep, Jordan.
How would you be able to make sure that they're
Depends a little bit on what thing we're talking about in terms of the biochemistry of how you do it, but there would be some biochemical way to set it up no matter what we're talking about. You call your chemist friend is how you do that. All right, any questions about this? Does this make sense? So yeah, as I said, that's one question that sometimes students are baffled by when they see on exams, and so that's how you deal with it. I also want to talk a little bit about issues of kinetics in a B cell response. If you look at today on the schedule, it says we're going to talk about kinetics and memory, and we're going to probably start into some of the kinetics and memory stuff. We won't get all the way through it because I really like talking about kinetics and memory. It's one where I can really go. But the end of the B cell slides I had for you were actually the beginning of B cell kinetics and memory, so everything kind of fits together. So first we're going to talk about this whole process with B cells, and then we'll apply it to thinking about T cells as well. So one thing that I want you to recall about an adaptive immune response is that we do have a primary and a secondary response. In a primary response, when we look at antibody production, there will be a bit of a lag before antibodies are generated. And at the beginning, the vast majority of our antibodies are going to be of the IgM isotype. And that's because that doesn't require class switching or a germinal center reaction. So here we can get cells proliferating, starting to make antibodies, but they won't have had to class switch. Later on in the response, we'll start to get IgG dominating. Because IgG is one of the isotypes that requires a class switch along with IgE and IgA, and IgG just happens to be the one that gets made at the highest level. So it's the one that ends up dominating. And this is all called the primary response. You can also notice on the right-hand side that, so this blue line here is the second immunization. So right now we're just looking at before that. At the beginning, we have mostly IgM. At the end of the time, IgG eventually catches up. At the beginning, we have mostly IgM. Eventually, IgG catches up. And you can see that the IgG affinity is getting higher too because of affinity maturation starting to happen a little bit late in this response. If we see that antigen again, either in the case of immunization, as is done in experiments, or if you get infected with it again, as in real life, then we have a secondary response. And the secondary response is different than the primary response in a number of ways. So first of all, we don't see that lag in when the response starts to happen. So the response is happening faster. You can also see that the response happens with a much larger magnitude. So we have a difference in the cells that are responding here, both in terms of quantity and in terms of quality, because they're able to respond to a much higher level, and they're able to respond much faster. And now our B cells making IgG are going to dominate. We are largely going to get IgG production. We'll get a little IgM production, and you'll notice that that IgM production is less than we had before. You can see the same thing in the figures on the right. After second or third immunization, the IgG numbers go way up. IgM doesn't really go up at all, doesn't change much, and IgG gets much higher affinity because of the affinity maturation process as opposed to IgM. This is just another way to do that comparison. The primary response is usually smaller, the secondary response is larger. The primary response is dominated by IgM. The secondary is dominated by IgG. antibody affinity in the primary is lower, antibody affinity in the secondary is higher. And in fact, we get the nicest secondary response to a protein antigen because we can get that nice help. Unless, of course, our other immunogen was linked to a carrier. So the same issues are being shown here. We can do lots of experiments to examine this process. And one such experiment is shown here. Here we can look at the responses made by 2 strains of mice. One strain of mice has a wild-type copy of AID and a mutant copy of AID. The other strain has no AID. So we've got AID deficient and AID sufficient mice. And the AID minus mice are in the opens, the AID plus mice are in the filled. And so first we can look at the IgM response to immunization. We've got immunization 1 and immunization 2 here. And if we look at the response to the first immunization, the mice don't look too terribly different in terms of the amount of IgM they're making. If we look at the response to the second immunization, maybe if you really wanted to push it, you could say, oh, look, the AID pluses have a little less IgM compared to the AID minus. But of course, AID, if you recall, was really important for both somatic hypermutation and for class switch. So if we look at a different isotype, IgG, that's important for class switch, we see a big difference. At the beginning, the 2 mice start with the same amount of antibodies, which is good because they haven't been immunized yet, so they shouldn't have big differences. However, quickly after that first immunization, the IgG response increases in the AID sufficient mice. The mice without AID don't make IgG, and you can see this becomes very dramatic by the time of the second immunization. And you would see similar data if you looked at CD40, CD40 ligand, anything like that. If you look here, you will notice that in the IgM plot, at the end, after second immunization, the AID plus mice are making a little bit less IgM. You saw that in the previous slide too. At the second response, the IgM level is in fact a little bit lower than it was at the primary response. This is an important part of the kinetics of a B-cell response. So let's think for a second about a B-cell response in terms of a primary and a secondary response. At the beginning, we will have a naive B-cell before antigen exposure, before the first infection. And one thing I love about this figure is it does not show a 0 on the y-axis. It shows that you have a little bit of B cells, but not 0, and that's important. So we've got a B cell. When we get infected, that B cell is activated. We get proliferation. That B cell differentiates to make plasma cells. Some make IgG, some make IgM. At the beginning, this naive B cell was only making IgM, and we'll have some contraction of these cells. Now, in this time where between your infections, there are a couple things you can think about. One of them, it's entirely possible that every single naive B cell responding to this antigen did not respond in that first go round. Why that's important is that it's possible that there's a naive B cell hanging out here too at this time. There was a naive B cell that did not get activated this first infection, and it's still chilling and it's present at the second infection. The other thing you can imagine is that VDJ happens all the time. And so even if that didn't occur, it's possible you could have made a new naive B cell. And there could be a naive B cell, again, hanging around, chilling at this time of the secondary infection. If we look at this process, when we get a repeat infection, we see responses being made by our memory B cells, our B cells that have switched to IgG. They're going to make that response fast. They're going to differentiate fast. They're going to make huge amounts of antibodies. They're going to be memory cells. but we really don't see a response of any naive B cells that happen to be around. In fact, the response is set up so that our naive B cells are turned off during secondary responses. So in fact, if there are any naive B cells around, they get turned off, because we don't want to waste cytokine on them. We want all our cytokines going to the memory cells, who are going to make the really good responses. And this process happens through a mechanism that's shown on the right. At the beginning, we have naive B cells. Naive B cells are shown in yellow. Our naive B cell is going to be able to bind to pathogen. There are no antibodies bound to that pathogen yet because the B cell hasn't made them. And so we're going to activate that B cell, see it gets a positive signal, and produce some antibodies. However, this B cell, a naive B cell, also has another receptor. It's an Fc gamma receptor. What does an Fc gamma receptor bind? Fc gamma, what's Fc gamma? So yes, Fc gamma is what it binds, but what is Fc gamma? Yeah, Emilio?
IgG.
Yeah, it's the Fc portion of IgG, the Fc part of IgG. Our B cells also have this Fc gamma receptor. If we are late in a response, there will be some IgG produced to that pathogen, and that IgG will bind to the pathogen. Now, when our naive B cell gets turned on, we will get a signal through both the B cell receptor and through the Fc gamma receptor, which will be ligated by the IgG that's bound to the pathogen. Our B cell is now going to get a negative signal, and our B cell is going to stay off. So that's going to keep our naive B cell off. Our memory B cell doesn't have that Fc gamma receptor, so it doesn't get an off switch if there are pre-existing antibodies around. But our naive B cell does get an off switch when that pre-existing antibody is around. You can see this exact thing happening here. So if we have our antigen, if it's bound to an antibody, that can bind the Fc gamma receptor, and that leads to a block in B cell receptor signaling. And so this is one way that we can make sure that only our activated B cells are responding in later responses and actually turn off our naive B cells. And that is incredibly important for many parts of this response. Does that make sense? We get this? Okay. Also, just as a preview, we are going to talk about a drug called Rogam a little bit later in the semester. And you're going to see this exact figure when we talk about Rogam, because this is actually how Rogam works too. It works using the same principle. So in general now, we want to think about the primary immune response as well as some general phases of immune responses. So recall with our primary immune response, we have a naive B cell or T cell present at the beginning. That shouldn't be 0. That should be a teensy bit above 0. That cell is going to undergo clonal expansion, differentiation, finally get rid of our antigen, and then contract before making a memory response. And you can see that process both in a figure that I drew and a figure from your textbook here. So what you can notice is that at the beginning, we're going to get infected with our virus. That virus is going to replicate for some period of time, as is shown in pink. Here, it's shown in black. So the virus is going to replicate to a high level. It's going to take a little while for that virus to do something to you that makes you feel sick. So shortly after the virus occurs, you're going to start to feel illness. That's probably based on the level of virus that you have. It's waiting until the virus is at a certain level before you get sick. Your primary immune response is going to be initiated. It's going to take a little while. Once we have a high-level primary immune response, we're going to clear that virus. Shortly after that virus is cleared, you're not going to feel sick anymore. And you can see that over here as well. At the time where we have high levels of CD4 and CD8 positive cells, the virus number comes back down. you're going to feel better, and you can see that eventually your primary immune response will contract as well of both your CD4s and your CD8s, though they contract at slightly different time phases. The first thing, of course, we want to address is why is there this lag? What takes so long? Because that's when we feel sick, and we want to not feel sick because no one likes that. And the answer is a lot of stuff. So we can see the fact that we need an innate immune response to turn on the adaptive response. We need interactions between APCs, dendritic cells, T cells. We need co-stim. We need trafficking, all of those types of things, as well as our proliferation, our differentiation, et cetera. So we've got a lot of events that have to happen. But as I stated before, really self? Awesome. One of those things that has to happen is this process of differentiation. With differentiation, our naive cell, our either naive cell, like T cell, or our naive B cell, is going to become a more specialized cell type. This could be an effector cell. An effector cell can get rid of pathogens. It has the effect. It can do the effector function. So this might be a naive CD8 T cell becoming a CTL. This might be a naive CD4 cell becoming a helper T cell. This might be a naive B cell becoming a plasma cell. Some of those cells are also going to become memory cells that are going to act later on. This slide should not be there. So we can think about what the difference is between, first, naive cells and effector cells. So a cell that is naive versus a cell that has been activated. One of the big differences is that our effector cells tend to be much more short-lived. It tends to be much easier to push them to apoptosis. They are very specialized to do a specific function. That actually is sort of stressful on the cell. Basically, the cells do their function and then they don't live for much longer. We can also see big differences in activation of different proteins. So for example, activated cells have 100 times more FOS than non-activated cells. They have 50 times more NFAT. They have 1,000 times more IL-2. They have much more of some of these adhesion molecules. And so effector cells are specialized to be producing a lot of cytokines, to be active really easily. And so our effector cells are going to work really, really nicely. But as I said, our effector cells are quite short-lived. And so the other cells are going to be the cells that are important for protection a long time later. We can see a lot of different types of responses that may happen following our initial infection. In the short term, after our initial infection, we may still have effector cells and antibodies around. So if you get sick and then your little brother gets sick, like a week later, you might still have high levels of effector T cells and antibodies. You might get that virus infection right away, and you might have such a great immune response that you shut it down immediately. So basically, you won't really see any type of replication or infection that might be referred to as an abortive infection. But if we go out longer, we will see a secondary immune response that can happen. And now what you'll notice is there is no lag in when that response occurs, and that response also is going to occur at a much higher level. And the idea of that is sort of shown here. So you can see our primary response. You can see that after the primary response, there has been contraction. The contraction has given us a higher level of cells than we had at the beginning. So there is a numbers difference here. but those cells are also qualitatively different. So when we get infected, the virus does get in. The virus does replicate. But your immune response expands so quickly that it's able to block that replication right away. And so instead of having this length of virus infection, you have a really short-term virus infection. You probably have a smaller magnitude virus infection. And because of both this short period of time and that low magnitude, you never feel sick. The virus wasn't around long enough to actually lead to feeling sick. The virus wasn't actually going to a high enough level to make you feel sick. This is the reasoning behind the idea that you can't get the chicken pox twice. You can get it the second time. It's just you're never going to notice it because your immune system gets rid of it so quickly and so well. This is the point where somebody always raises their hand and tells you about their cousin who got their chicken pox twice. But this is the idea of why previous infection will lead to protection, because your primary response is going to lead to this nice protective secondary response. This secondary response varies from the primary response in a bunch of ways. At the primary response, you're starting with a small number of cells. You're starting with a larger number of cells in the secondary response. In the primary response, there's a delay before you have antibodies. In the secondary response, you've already got some antibodies. Class switch has happened in the secondary response, as has affinity maturation. Not so with the primary response. And in the secondary response, the adaptive and innate immune systems are working much more closely together. I really don't like the order of these slides. And so you can see that, first of all, in these antibody responses. So if we look at a person making a primary response, they have between 1 in 10 to the 4th and 1 in 10 to the 5th B cells specific for an antigen. You can see that's much higher. In a secondary response, you can see the big differences in class switch. You can see the difference in affinity. You can see the difference in hypermutation, all as a result of that secondary response. But we can also think about one other question about these memory cells and these memory responses. I told you that the effector cells tend to be relatively short-lived. In fact, frequently they are now referred to as SLECs, or short-lived effector cells, in the literature. But what I didn't tell you about is how long the memory cells live and what goes on with the memory cells. People have spent a lot of time trying to figure out the answer to this question. I'm going to tell you what the slide says. I'm going to tell you what one paper says. That paper is a little bit controversial, but I'll say that paper gives us an idea of the range. So here we can actually look at people who got a smallpox vaccination. The smallpox vaccination has not been given in the United States since the 1960s. And so we can actually, and smallpox is not infecting people in the United States. So if you have a smallpox secondary response, if you have IgG against smallpox, we know that's from your memory cells that have lived for a long time. And we can watch the antibodies to smallpox, the CD4 and the CD8 memory cells, decline in response to smallpox. And what you will notice is that more than 30 years after vaccination, we can still detect high levels of antibody, high levels of CD4, high levels of CD8. We can see that we can detect antibodies in vaccinated people out to 75 years following vaccination. And you can see that with antibodies at the top, T cells at the bottom. The 75-year number, like I said, is a little bit controversial. You can imagine it's probably pretty hard to do those experiments because you have to find people who got vaccines 75 years ago who have a record of that, who are also still around and are willing to give you blood and let you do this experiment. It's not an easy thing to do, but the data suggests that memory cells can live, or at least can be replicating and producing progeny out to at least that long following infection. And so these cells are quite different than our effector and naive cells, and we will talk more about those differences and some super exciting stuff on Wednesday.